Thank you for having me, and I'm very happy to be here today to talk about the bit of a long name, Danish BioImaging Infrastructure Image Analysis Core Facility. And we've been in operation for about 2 years now, so I'd like to share with you some lessons that we've learned from our first 2 years. So just to kind of explain why the long name. So you've heard today this morning from Thomas from Danish BioImaging, and Danish BioImaging is the Danish node of Euro-BioImaging. So we received funding in 2021 from the Ministry of Higher Education and Science, yes, to bring these multiple facilities across Denmark and allow the life science researchers to access equipment and expertise across the country. And so this infrastructure supports communication between these different open access facilities. And so we have a facility access portal, a website where you can see all the different facilities that there are, what resources that they have, and how you can get in contact with them. Social media run by Sonia at the back over there taking pictures. And a course catalog, and it's also even got like a cross-institutional course. So the students go around the different institutes and take a course together in that sense, right? So the infrastructure also coordinates investments to implement new bioimaging technologies. I'm not going to get too deep into all of this. If you want to know more, talk to Thomas Nuno. They have a poster that will let you know more about this. So the third thing is that this infrastructure also is there to include an implementation of such a nationwide open access bioimage analysis service, which is us, the Image Analysis Core Facility. I'd also just like to quickly draw your attention to the fact that there are computer science groups that are part of this infrastructure. And I think that kind of really highlights what this Image Analysis Core Facility does that is kind of separate and fills in that space over there, right? So our mission is to provide open and remotely accessible services to support life scientists and medical researchers in doing their analysis, visualizing their data, extracting quantitative information from their data sets. So we're here to bridge that research gap between computer vision scientists and the existing life science researcher user base. Right, so we're here to support the work of the already existing facilities that may not have a locally embedded image analyst. Some of them do, many of them don't. And finally, we're here to promote the knowledge between these facilities as well. So that's the team and current and past. Right. There's a little pop-up on here that wants to. Sorry. Okay, now I'm going to stop it. All right. Computer issues, right? Okay. So to fulfill this mission, we have a facility model of our services, and it looks like this. We have essentially 3 sections that we focus on. So that's the knowledge of it. So here we have teaching, where the workflows are, building with repositories of our tools. We have communication, where we're trying to link the facilities up, network outside of that, and also just foster more collaborations, right? And we also have infrastructure in terms of data exchange and providing computing solutions. More concretely, that translates into, for teaching things like training, workshops, and courses, and actually working with the different facilities. I'm going to focus this talk more on the last 2, this bioimage analysis consultation service and the project services that we provide, and the infrastructure that we have, right? So not sure, but I think attendees in 2023 may have had a talk from our then facility head, Sebastian. And so it's been 2 years since we started, right? I'd like to share our experience of implementing these actual actions, how that's worked out for us, the lessons that we've learned from it, and some of the future plans that we will be embarking on. And yes, I'll largely be discussing this last 2 over here. Right, so infrastructure, you start a new facility, first thing you do, you go out and buy some machines, right? So we started out with 3 workstations that we put online and made them publicly bookable and remotely accessible, because like I said, we are also trying to serve users from across Denmark. And then we load it up with your usual open source software like Fiji, we've got Nepali, the normal stuff, right? And so since we also share an office with CFIM, the core facility for integrated microscopy at KU, we also connect users with the software licenses that exist there. So we shared the Imaris license with them, and through KU, we use the MATLAB license, and that's that. So apart from the local infrastructure, we also make use of the Danish national infrastructure, because there's quite a lot of initiative going on to build such national infrastructures for computing in Denmark, especially through the Danish e-infrastructure consortium. So we use our cloud computing right now via SDU's uCloud, so that's the South Denmark University. And we also right now use our storage with the University of Copenhagen's Electronic Research Data Archive, which will be the basis, as far as I understand, of a future national level storage and sensitive storage facility, right? And our services, which is really the core of what we provide to users, we began with 4 pilot projects in April of 2023, and finally opened for service in September. And the user workflow basically goes something like this. They submit a project to us, we review it, we see whether or not we can do something about it. And if not, we either tell them to kind of refine what they're trying to ask for, or we ask some third parties, like we said, we work also closely with the computer science departments, and sometimes the projects are a bit long, so we push that towards them. And the main deliverable for us being at the end a workflow, and then we also teach the users how to use it. And that's kind of like an iterative process. We make a workflow, we teach them, and they say, oh, okay, can you change some things about this? This thing's not working quite right for me. And then we go back and we do that until we get a point where we're satisfied with where it's going. We're satisfied with where it's at, and they're happy with it. And then we kind of do the normal archive it, publish it, put it in storage, and all the cleanups and stuff. And that's where we're at, right? So since we started, I'd just like to give some project statistics and highlight some of the projects here. So our user distribution, over the course of this 2 years, we've managed to get actually quite a representative usage from across the Danish facilities, in the sense of it's roughly to a scale of how many facilities are in each location. And this actually took deliberate effort, because at the start, it was very easily skewed towards basically where we were at. So we actually had to reach out and, to these other facilities and talk to them and try to get more users from their side. And I'll tell you a little bit more about it later as well. And also in terms of project lengths, we had to also learn to manage what kind of projects we can't really do. So there's a range of applications that we get. So some are quite simple. Some are like, please, you can solve it by saying, here's this tool. You can go ahead and use that one. And there's some tutorials that are already available. So that's quite all right. And then some of them, there's, long 70-hour of like coding kind of projects where it becomes, okay, this is a bit too long now. This can be a whole research project for an entire PhD student, right? And so it would be unfeasible to do that at a facility. And so as image analysts, we have the kind of experience to say that, okay, maybe this is more complicated than you expect. You expect that you go to a facility and they can fix it, but maybe not. And then at this point, we redirect them to other people that we know might help. And like I said, this is a skill that took us some time to learn, and also communicate with the users very realistically about what can or cannot be done, right? And it even came up during our pilot project phase, so we're very aware of this. So we find that somewhere in between that is where we can actually provide image analysis service as a service quite meaningfully. So that's things like tricky data sets where your out-of-the-box solutions just doesn't work for you. And then you have to do a bit of like fine-tuning and all of that for your specific data. And also things like, I have over 9,000 images. Please help me automate a workflow. I can't do this by myself by hand anymore. And that is kind of the sort of service that we expected to provide when starting custom workflow services. So here are some examples of that below. So some things, cell segmentation, where the data was quite similar to it. Okay, so there's a lot of them. I'm just going to go through, I think, maybe just one. Yes, so for instance, there was this one right at the leftmost side for you guys, where we're trying to segment some neurons from essentially, some histochemical staining. And they're using QPAR for it, but that's not very easy to do the measurements. So we kind of built that Fiji bridge on their behalf, right? So these are the kind of standard projects that we get. So, but we also have had some projects from our applications that really expanded our notions of what services we can offer, and I'd like to highlight those next. We've had requests for code review and workflow optimization. I didn't realize that that was something that we could provide, but it is. So this example that we have here is now a paper, and it's a food science group at SDU that wanted to apply cell pulse essentially iteratively at different scales so that they can segment their essentially these kind of foam bubbles so that they can get the texture of this food that they are looking at, right? And then they came to us with their workflow and said, it's a bit slow, can you help us optimize it? And what are we doing wrong here? So, we just looked through it and we gave some feedback suggestions. Here's where you can reduce some of the for loops. Here's where you can go and use a library that already exists. Things like that. And yeah, we didn't realize that we could do that. And now we've also had more projects come in that look a little bit like that. So that's quite exciting. And also another one is to entirely replicate published workflows. So in this case, a user had read a paper and wanted to apply the method in exactly the same way. Unfortunately, that paper was published without any code, even though it had a detailed method description. So, like the reproducibility crisis and all that, right? But as image analysts, we could understand the methods that were being applied. So we could turn that from a written description of the method back into a code that could be used. Right, so that's quite something that I wasn't really expecting, but you can also hear more about this at the Image Data Community Days, where we'll be talking about, two of my colleagues will be talking about it, turning research back into user-centric tools, right? So finally, we have our consultation services, which for our BIA workflows, the users need to submit an application form, which is not long, but it is quite specific in asking for the type of help that they needed. And they need to know what they want to have measured, all of that kind of things. And sometimes users just don't know what they need. So we have a regular call for help to lower the entry barrier as a starting point for the users. And we also actually found it really helpful to direct users to the appropriate amount of support that they need. And we find that the users that have had consultations end up with more successful projects, where successful kind of means that they had a better expectations of how things are going to turn out, right? And that they're getting the right level of help. So some of our learnings and the future plans from it, the first one being that people who come to us sometimes get confused. They don't know whether they're here for like a consultation, whether if they just get a computer and some training, they'll figure out what they need. So we're planning to just simplify our points of entry and direct more people towards the consultations so that they can, have some of the help before they start requesting services. And other thing is that beginnings are pretty slow. So when we first started out, so that's the one on the right, left call for help, we spent our first 6 months when it's open, literally nobody, crickets, silence, right? And we are wondering, why is nobody signing up? Is this even worth doing? We tried promoting it everywhere, and we know that there's unmet needs, but where is everybody? So and we talked to people after that, and we start reaching out for help and stuff. But sometimes it's just that patience is key. It takes a while for news to spread about the services being available. We actually talked to another group recently where they said they tried this call for help thing, but after 6 months, they also got no response in the whole time, so they shut it down. I think we shut it down in the first 6 months. We would not have any of the services that we had here. So it's okay to take things kind of slow and easy. And another thing in terms of marketing or reaching out, as we were talking about earlier today, is to work with the local facilities. And that's where we found our most effective outreach. Because what really turned things around was that when we had a physical presence and we collaborated with the other facilities, that actually translated into users for us. So people who hear about us mostly have attended our courses, for instance, or have were referenced in like redirected in person by a facility staff, right? And yeah, so it took a while. So it's more like in our second year when we started reaching out towards other places. We started holding workshops there that we managed to kind of skew our balance of user access towards being more balanced between the different facilities in Denmark. So here, from before, I think one of the things that was quite poorly defined at the start was this communication. We had this idea to link facilities, network, collaborate, but what does that mean concretely and tangibly, right? So I'm going to wrap up some of the ways in which we are more tangibly trying to build these connections. So teaching is, of course, one of them. We've started now teaching, bringing our courses or actually being invited to courses at different nodes in the Danish BioImaging infrastructure community, right? And that has really helped us in the past, and we really would want to continue doing this. I think it benefits the students at the other side and also the facilities there, and really raises knowledge. Another thing that we've been doing is based on our ongoing call for help, where we bring ourselves to the facility and just are there for a day. And we have a physical open office there so that people can see us, because right now we do it online. And I think that kind of creates a barrier in which people, they're unfamiliar with this. They've never seen these people before. They've never seen us before. And so being physically there, I think makes us more approachable. Right, and this also brings us into deeper collaboration with the other facilities, because now we're not just people who attend the same conferences together. We are people actually working together towards a goal and making something happen. And I think that's really nice. And so, yeah, we've had two now. I think the first one was really successful with us attending to about 12 participants in about 4 hours, so 20 minutes each. Just ask your questions. We'll try our best. We're not going to open up the computer and solve it right there, but that's okay. You have some advice, right? And finally is to innovate and build better connections also to the computer science department that's there. So as I said, there are essentially very compute heavy nodes in the infrastructure, and we already take part in their meetings through their image section at the computer science department, and also with DTU Copenhagen through like this Center for Quantitation of Imaging Data, KIM, right? And what because we attend these meetings, we had this idea that after the meeting, let's put some time aside, have a hackathon to try out all these new techniques that we've heard about at the meeting. So we've had our first one just a couple of weeks ago, where we tested software that was developed in the section. So here we tried Root Painter, which is essentially a UNet with a nice interface in front of it that's already being used a little bit in the plant community. And then we also had some talks from the imaging, the computer vision scientists that are working there. And I think this was one of the very interesting ones that I saw, where they were using essentially diffusion models to break the symmetry of segmentations, like from CNNs and from cell posts and things like that, and to be able to essentially segment overlapping cells, which was, oh, that's really cool to me. But that's not really something that's widely already available to use, but it's something that is, okay, we're trying this thing out, but it's so slow that it doesn't seem to be a thing that is ready to use for users. But we're keeping on top of things. And in the interest of connecting, it would be happy if you would like to connect with us. You can contact us via our email, website, our social media. And thank you.